Hey there, I am Maruk Said, a microbiologist and a science educator. Science is my passion and I'm here to share it with you. Here are some definitions of microbiology and molecular genetics. Before starting this video, don't forget to subscribe to my channel and like this video. Let's learn about microbiology, all relevant definitions. Number 1. Microbiology. It is a scientific study of microorganisms, which are microscopic living organisms that include bacteria, viruses, fungi, archaea, and protozoa. Number 2. Bacteria. They are single-celled prokaryotic microorganisms that have a wide range of shapes and sizes. They can be beneficial, neutral, or pathogenic, playing essential roles in various ecosystems and human health. Viruses. They are non-living infectious agents consisting of genetic material, DNA or RNA, enclosed in a protein coat. They require host cells to replicate and can cause various diseases in plants, animals, and humans. Fungi. They are eukaryotic microorganisms such as yeast and molds. They play a crucial role in decomposition and nutrient recycling in ecosystems, but some species can also cause diseases in plants and animals, including humans. Archaea. They are single-celled microorganisms with characteristics similar to both bacteria and eukaryotes. They can inhabit extreme environments like hot springs, acidic conditions, or deep-sea hydrothermal vents. Protozoa. They are unicellular eukaryotic microorganisms that often possess animal-like behaviors. They can be free-living or parasitic and are commonly found in aquatic environments. Pathogen. A pathogen is a microorganism, such as a bacterium, virus, or fungus, that can cause disease in a host organism. Antibiotic. Antibiotic is a substance produced by microorganisms, typically bacteria or fungi, that can inhibit the growth of or destroy other microorganisms. Antibiotics are widely used to treat bacterial infections in humans and animals. Antimicrobial resistance. Antimicrobial resistance, AMR, occurs when microorganisms develop the ability to survive the effects of antimicrobial drugs such as antibiotics or antiviral medications. It is a significant global health concern as it can reduce the effectiveness of treatments for various infectious diseases. Sterilization. Sterilization is a process of eliminating all forms of microbial life, including bacteria, viruses, fungi, and spores. It is commonly used in healthcare settings and laboratory environments to prevent contamination. Vaccination. Vaccination, also known as immunization, is the administration of a vaccine to stimulate the immune system and provide protection against specific infectious diseases. Vaccines are made from weakened or killed pathogens or their components. Culture medium. A culture medium is a nutrient-rich substance or environment used in a laboratory to grow and cultivate microorganisms outside their natural habitats. It provides essential nutrients for microbial growth and reproduction. Colony. In microbiology, a colony refers to a visible cluster of microorganisms that have grown and multiplied on a solid culture medium. Each colony typically originates from a single microbial cell. Pathogenesis. Pathogenesis is a process by which a microorganism causes disease in a host organism. It involves the steps and mechanism through which a pathogen invades, multiplies, and damages the host cells or tissues. Antibiotic resistance. Epidemiology. It is a study of the distribution and determinants of health-related events such as infectious diseases in populations. It helps identify patterns and risk factors associated with disease outbreaks. Virulence. Virulence is the degree or intensity of pathogenicity exhibited by microorganisms. It indicates the ability of a pathogen to cause severe disease in a host organism. Biofilm. A biofilm is a community of microorganisms such as bacteria that adhere to a surface and form a protective and often slimy matrix. Biofilms play an essential role in various processes such as water purification and medical device-associated infections. Antiseptic. An antiseptic is a substance or a chemical agent used to inhibit the growth and activity of microorganisms on living tissues. They are commonly used to disinfect skin or wounds. Disinfectant. A disinfectant is a chemical agent or physical method used to destroy or eliminate microorganisms on inanimate surfaces and objects, reducing the risk of infection. Opportunistic pathogen. An opportunistic pathogen is a microorganism that typically does not cause disease in healthy individuals but can cause infection or disease in people with weakened immune systems or other predisposing factors. Endospore. An endospore is a highly resistant, dormant structure formed by certain bacteria as a survival mechanism when environmental conditions become unfavorable. Endospores are highly resistant to heat, chemical, and desiccation. Antigen. An antigen is a foreign substance, such as protein or polysaccharide, that can trigger an immune response in the body. It can be recognized by the immune system, leading to the production of antibodies or activation of immune cells. Antibody. An antibody, also known as immunoglobulin, is a Y-shaped protein produced by the immune system in response to the presence of antigens. Antibodies help in neutralizing or eliminating pathogens from the body. Zoonosis. Zoonosis is an infectious disease that can be transmitted from animals to humans and vice versa. These diseases play a significant role in public health and can emerge through close contact with infected animals or their environments. Kindly show your support by subscribing to my channel. Prophylaxis. It refers to the preventive measures taken to protect against development or spread of infectious diseases. It may involve vaccinations, the use of antimicrobial agents, or other preventive strategies. Nosocomial infection. It is also known as hospital-acquired infection. It is an infection acquired during a stay in the healthcare facility such as hospital or nursing home. A bacteriophage, or phage for short, is a virus that infects and replicates within the bacterial cells. Bacteriophages have been studied extensively for their potential use in treating bacterial infections. Microbiome. The microbiome refers to the collection of microorganisms including bacteria, viruses, fungi, and other microbes that inhabit a particular environment such as human gut or skin. Binary fission. Binary fission is a method of asexual reproduction in which a single-celled organism such as bacteria divides into two identical daughter cells. Conjugation. It is a form of horizontal gene transfer in bacteria where genetic material is transferred between two bacterial cells that are temporarily joined together. Transduction. It is another form of horizontal gene transfer in bacteria where genes are transferred from one bacterium to another through a bacteriophage, a virus that infects bacteria. Transformation. It is the uptake of foreign DNA by a bacterial cell and its integration into the recipient cell's genome, leading to genetic change. Acid-fast staining. It is a laboratory technique used to identify bacteria that have a waxy cell wall such as Mycobacterium tuberculosis, which causes tuberculosis. Nitrogen fixation. It is the process by which certain bacteria and archaea convert atomic nitrogen N2 into a biological usable form such as ammonia or nitrate. This process is essential for nitrogen cycling in ecosystems and provides an important source of nitrogen for plants. Bioremediation. It is a process that uses microorganisms to break down or remove pollutants from contaminated environments. It is an eco-friendly approach to clean up environmental pollution. Quorum Sensing. Quorum Sensing is a system used by some bacteria to communicate and coordinate their behavior based on population density. It plays a role in regulating gene expression and viral infectors. Viral replication. It is a process by which viruses multiply and produce new viral particles within a host cell. It involves various stages including attachment, penetration, replication, assembly, and release. Bacteriostatic. Bacteriostatic agents are substances that inhibit the growth and reproduction of bacteria, preventing them from multiplying without necessarily killing them. Bactericidal. Bactericidal agents are substances that kill bacteria outright, leading to their complete destruction. Viron. A viron is a complete infectious viral particle consisting of nucleic acid, DNA, or RNA surrounded by a protein coat called a capsid. Phage therapy. It is a therapeutic approach that uses bacteriophages to target and kill specific bacterial pathogens in bacterial infections. Probiotic. Probiotics are the live microorganisms, usually beneficial bacteria that, when ingested in adequate amounts, confer health benefits on the host by improving the gut microbial balance. Fermentation. It is a metabolic process in which microorganisms, typically yeast or bacteria, convert sugars into alcohol, acids, or gases, often used in production of food and beverages. Toxins. Toxins are harmful substances produced by certain microorganisms that can cause adverse effects on cells or tissues of the host. Capsule. A capsule is a protective outer layer found in some bacteria and fungi. It can play a role in evading the host's immune system and contributing to the pathogenicity of microorganisms. Lysogeny. Lysogeny is a state in which the bacteriophage virus that infects the bacteria integrates its genetic material into the host bacterium genome and remains dormant without causing immediate cell lysis. Bioinformatics. Bioinformatics is an interdisciplinary field that combines biology, computer science, and mathematics to analyze and interpret biological data, particularly genomics and proteomics data. Microbiota. Microbiota refers to the community of microorganisms that inhabit a particular environment such as human gut, skin, or soil. So that's it for today. Follow my next video for Microbial Genetics definition. Till then, don't forget to like this video, subscribe to my channel, and press the bell icon button so you don't miss any of my videos.